Hey guys, welcome to another video on microbiology, which deals with the classification of microorganisms. In the previous video, we studied about the history of microbiology, and in this video, we'll be studying about the various classifications that are seen under microorganisms. So, moving to the very first slide, the microorganisms can be classified into prokaryote and eukaryote. So, what is prokaryote and eukaryote? We'll be studying in further part of the video, and now we'll see what are the classification, just the general part. So the major groups of microorganisms that can be observed are bacteria, archaea, protistas, fungi, and viruses. So these are the five main microorganisms that we are going to be studying in this video. So firstly, what is prokaryote? Prokaryote is a small unicellular organism that do not have a well-defined nucleus or any specialized membrane-bound organelles. So these prokaryotes do not have a defined nucleus. A defined nucleus is a nucleus which doesn't have a nuclear membrane. So the nuclear material isn't bound by any nuclear membrane, and a specialized membrane bound organelles like mitochondria, ribosomes, or the Golgi apparatus aren't found in prokaryotes. So the prokaryote cells are usually measure about zero point 0.1 to 5 micrometer in diameter. They have cell wall, plasma membrane, and cytoplasm. Some prokaryotes have pili, flagella, and fimbriae, which are the locomotory organs, which help in conjugation, movement of the body of the bacteria. So these prokaryotes might contain ribosomes, which are present in few species of prokaryotes. So under prokaryotes, we'll be studying about bacteria and archaea. Firstly, we'll see what is bacteria. Bacteria is very ubiquitous. Ubiquitous is commonly found everywhere. They inhabit a variety of ecosystems and can tolerate extreme conditions like those of a fauna that is found in toxic waste, Arctic ice, and hydrothermal vents also. So these can tolerate extreme conditions, which are toxic waste is nothing but the uh, nuclear or the chemical waste that is being found. Arctic ice is the freezing cold, that is cold temperature, and hydrothermal vents. Hydrothermal vents is the steam water that is found in certain places on Earth. And these bacteria are tiny single-celled organisms that have variety of shapes. They can be spherical, they can be spiral, comma shaped, corkscrew shaped, or rod shaped. So I've put an image of it. So you can see there is Staphylococcus aureus, Clostridium butylii, which is rod shaped. Klebsiella is also rod shaped. Clostridium tetani. Then we have Streptococci, which is spherical. Then we have a rod shaped Bordetella. Then we have Neisseria gonorrhea, which is also spherical. Then we have E. coli, Vibrio cholera, also rod shaped, Streptococci. Then we have Bacillus, then Helicobacter pylori, and Treponema pallidum. These are the different bacteria that I have put into an image. So moving to the next part, these bacteria sometimes occur in chain. They might be in clusters. They might be in pairs or tetrads. So I've put the pairs. Paired bacteria are Neisseria and Streptococci and a few others. And there is a chain form which is Streptococcus, and there is a cluster also, that is Staphylococcus aureus. So these are the different uh, organisms which form a cluster, chain, or a pairs or threads. So these prokaryotes having no nucleus with the genetic information contained on loops that have no specific location in cell. So these prokaryotes have genetic material which isn't bound by any certain membranes or boundaries. So they float in the cell cytoplasm itself. So some bacteria also have certain circular strands of DNA called plasmids. You can see that in the image also. There are circular DNA which is plasmid, and these plasmid helps in replicating of the bacteria independently. And this plasmid sometimes confer advantages. There are advantages that sometimes, like they form a house of gene that makes the bacterium pathogenic or more virulent than other strains. So it helps the bacteria to survive in spite of any certain outside attack on the bacteria. So they are virulent, and bacterial cells are shaped by a cell walls made of peptidoglycan. So the cell wall is made up of peptidoglycan. They also have a permeable plasma membrane that functions in the transport of molecules into and out of the cell, and a capsule which offers a layer of protection for the cell. So there's a outer membrane which comprises of the capsule uh, capsule as well. So these capsules help in determining the capacity to cause disease from the bacteria. These bacteria can be flagellated. They have flagella, and these flagella act like a whip-like structure and help in the locomotion. There are certain hair-like appendages, as I told in the previous slide, the pili, sorry, it's pili. I told that certain prokaryotes have pili, flagella, and fimbria in this slide, right? So bacteria also may contain flagella or hair-like pili. So these help in the transfer of DNA from one bacteria to another. So, based on the cell wall composition, the bacteria is further divided as gram-negative bacteria and gram-positive bacteria, which we will be discussing in the upcoming videos. So, for this, in this video, it's just a general briefing of what is bacteria. Moving to the next part, which is Archaea. Archaea contains some of the oldest species on Earth. They are also known as extremophilus because they can withstand extreme environmental conditions. They're found in many places thought to be inhabitable by most other organisms. So this archaea can be classified based on environment of food source into Crina Archaeota, which is found in the environments with high temperatures such as hydrothermal vents and hot springs, and Urea Archaeota, which is organisms that grow in high salt environments. So they have these archaea have a very different kind of cell wall composition. They lack peptidoglycan basically. They lack this peptidoglycan, and they have a very different cell wall composition which helps in protecting the archaea from the extreme climatic conditions that it survives in. So these cell wall compositions and RNA sequences and coenzymes play an important role in forming the cell wall, a unique kind of cell wall. Then the archaea are more relevant with certain aspects with eukaryote. For example, they have similar mechanism of protein synthesis. So these archaea species are unculturable. They cannot be cultured. So that's the images that I've put for archaea. There is one such crania archaeata which I've put in the image, and Uri archaeata, the another image that I've put. So these are the archaea classification. Moving on, eukaryotes, the classification under microorganism. So eukaryotes are the microorganisms which could be unicellular or multicellular. They have a very defined nucleus which is enclosed in a genetic material, and they have membrane bound organelles which are mitochondria, chloroplast, endoplasmic reticulum, Golgi apparatus, and lysosomes. So these eukaryote microorganisms have a defined nucleus and a membrane-bound organelle. They could be unicellular or multicellular. So moving to the very first eukaryotic microorganism, which is Protista. So Protists are the unicellular eukaryotic microbes. Like all eukaryotes, they have nuclei and membrane-bound organelles. Protists were the first microbes visualized by scientists at the dawn of microbiology. So protist was the first microbe which led to the very big study on microorganism, which is microbiology. So one of the important protists is malarial pathogen, which is Plasmodium, has caused millions of deaths worldwide annually. Another is the vector transmitted Trypanosoma brucea that affects the animals and causes sleeping sickness in humans also. Protists are the very important in food chain. They act as the decomposers in the food chain by recycling the nutrients and contribute to the biomass in aquatic environment. So these are called as the uh, decomposers, which help in the recycling of the nutrients and help in contributing to the biomass of aquatic ecosystem. So these protists also help in production of Earth's oxygen through photosynthesis by symbiotic relationship. Symbiotic is mingling with other microorganism or the organisms, and together they produce certain products that is called as symbiotic relationship. Moving to the next part of our classification, which is fungi. So fungi are the eukaryotic organisms that may be unicellular or multicellular. They have a well-defined nuclei that house the genetic material and membrane-bound organelles. Then these fungal cell walls are not like peptidoglycans that we observed in the previous slides. So fungal cell walls have certain other composition which are chitin and glucons, and these cells do not have chloroplast and chlorophyll as plants do. So these fungal plasma membrane contain a sterol or ergosterol which acts to preserve its integrity, and fungi grow vegetatively as hyphae. You can see the image of a hyphae, and the fungi reproduce both sexually and or asexually. So many have more than one type of spores. So the fungi have more than one type of spores, while reproduction. Some fungi are dimorphic, changing their state according to the environmental condition. Some plant pathogenic fungi, for instance, have yeast and filamentous stages in the life cycle that function in dispersal and infection of the host. So fungi can be classified as zygomycota, deuteromycota, ascomycota, and basidiomycota. So these are the different classifications under fungi. The members of the group Oomycota have long been thought to be of true fungi, but they have filamentous growth and absorptive nutrition. However, they are not true fungi because they have different cellular composition. So this ends up the classification of fungi. Hope you like the video. Please to subscribe to the channel to get a more updates on the RJUHS syllabuses that I have been trying to upload very recently. Uh, Hope you like the video. Thank you.